Welcome to MrCScience.com's video on genetic recombination. In our last video, we mentioned that bacteria reproduce asexually. This means that the daughter is basically a clone of the parent. The genetic information is exactly the same. There can be coding errors, but as we'll find out later in the book when we talk about DNA and reproduction, coding errors destroy the DNA. Basically, they take out information. Sometimes this can be useful, but they don't. Coding errors don't add information. We've yet to observe any coding change or mutation in the DNA that's added information. But we do know that bacteria do mutate and gain new genetic codes. So how does this happen? This happens through genetic recombination. Now, the advantage of this is if a population is all the same genetic material, no genetic diversity, if something major happens that genetic material cannot work against, it can wipe out the entire population. So genetic diversity is very handy. You notice in no, that's a different thing we'll talk about. So how do bacteria go through genetic recombination? Well, there are three different procedures. There is conjugation, transformation, and transduction. We're going to talk about each of the three. The most complicated portion of this is conjugation. Again, I could draw this out for you, but your book has a very good picture of it on page 48. Figure 2.6 is conjugation in bacteria. So let's talk about what happens. First of all, conjugation does not exchange direct genetic material. Their DNA is not exchanged with the other DNA. They exchange what are called plasmids. Your book defines a plasmid as a small circular section of DNA that confers one or more traits to a bacterium and can be reproduced separately from the main bacteria's genetic code. So basically, it has a separate chunk of DNA that it can pass on to the other one. The bacteria will decode both and use both pieces of DNA. So if we look at the figure, so we got our plasmid inside of one of the bacteria, and it's uncertain what causes it to pass that on to the other bacteria. But first thing that happens is the fibria. The fimbria of two bacteria connect. These fibrium are now relabeled sex pili so that they can connect. The one bacteria with the plasmid duplicates that plasmid, and then a tube forms in between the two bacteria called a conjugation tube. Hey, conjugation tube. And the one new plasmid is passed through this conjugation tube. It enters the other bacteria. It is reformed into the plasmid. The conjugation tube collapses, and now both bacteria have this new genetic trait. The genetic trait is only passed one way, so it's not both directions. There is an exchange. There is not a daughter, so this isn't sexual reproduction. This is just conjugation that happens in bacteria. Another way bacteria can gain new information is through transformation. Your book defines transformation as the transfer of a DNA segment from a non-functional donor cell to that of a functional recipient cell. Basically, if a bacteria dies, another bacteria somehow recognizes a piece of DNA and brings it into the cell and creates a plasmid with it and uses it. That DNA may provide a new trait. It may just be existing DNA that already exists in the other bacteria. When reproduction or when asexual reproduction happens, it will not be coded, so it doesn't matter. But conjugation allows it to pass a trait between many bacteria. Transformation is a way to pick up traits off of a dead bacterium. Last one, transduction, is defined as the process in which infection by a virus results in DNA being transferred from one bacterium to another. Basically, what happens if you understand viruses, viruses are just bacteria coated in a capsule. That capsule attaches to a cell, injects or not bacterium, it's just DNA or RNA um, coated in a capsule. That virus attaches to the cell, injects that DNA or RNA into the cell, be it a bacteria, being it an animal cell, being it any cell, and that cell says, hey, I know what to do with DNA, and it decodes it and runs it basically. So, because of that transfer, viruses can also transfer genetic information from one bacteria to another. That's the last way to transfer those. So, conjugation, transformation, and transduction all can help diversify the bacteria's genetic code. Um, a lot's not understood about why this happens, but just remember conjugation is between two bacteria where one plasmid's transferred. Transformation is one bacteria where it takes in genetic information from a dead bacterium, and then transduction uses a virus to pass the genetic information. If you have any questions on this at all, just drop me a line at mrc@mrcscience.com. Thanks.